Alright folks, so this is the macromolecule proteins. Lots to talk about with proteins, so I made it its own mini lecture all by itself. So first of all, proteins are why your phenotype is your phenotype, or another way of saying that, it is why you look the way you look. And you're going to yell at me and you're going to say, no, it's my DNA. Well, it is. It is your DNA, but most directly it's your proteins. So what the heck do I mean by that? Well, your DNA dictates what your proteins look like. But your proteins are what dictate what you look like. It's your phenotype. Lots of different types of proteins. As you can see here, we got enzymatic proteins. We used to think that all enzymes were proteins. But by the way, not all proteins are enzymes. But we used to think that all enzymes are proteins. And then not too long ago, I think it was probably the 90s, we discovered some catalytic RNA molecules. We call them ribozymes. So now we know that not all enzymes are proteins, but 99% of them are. I mean, the ribozymes are a small portion of all these enzymes that we have. So enzymatic proteins. We have structural proteins. The structural proteins are a lot of connective tissues like collagen and cartilage and things like that. So we got structural proteins. We got storage proteins. I can't really think of a lot of storage proteins in humans. We do keep a nutritional, we do store some amino acids, but storage proteins are things like egg albumin, you know, the egg white, and when you crack an egg, the egg white. It's things like the casein that you find in milk, the protein casein. It's what makes the milk white, in fact. So humans make milk. We are mammals. We make milk. So I guess we do have storage proteins because we have milk proteins as well. Transport proteins. Humans have tons of these. We have things like transferrin, which transports iron, hemosiderin, hemoglobin, ceruloplasmin, transports copper. Just all kinds of transport. We have albumin's a big one. Albumin's the predominant protein in your blood, and it transports lots of things for us. So we got lots of transport proteins. We got a whole bunch of hormonal proteins. I would say probably most of our hormones are proteins. Really, I've never really sat down and did the statistics on that. But many, many, many of our hormones are proteins, like FSH and LH and growth hormones are protein, and insulins are protein. I mean, there's a ton of hormones that are proteins. Now, not all of them are. Some hormones are steroids, and some hormones are amino acid derivatives. But many, many of them are proteins. We have receptor proteins, huge. Receptor proteins is a huge class of proteins and extremely important. Why do I say extremely important? Well, your hormonal levels could be absolutely normal. But if you don't have the receptors for that hormone, then you have a disease. So see, it's not just having the proper signal molecule, you must also have the receptor for it. We have contractile proteins. That's the actin and myosin of your muscles. It's what makes you strong. We have defensive proteins. These are called immunoglobulins or antibodies. It's also called immunoglobulins. Immunoglobulins. We also sometimes call them gamma globulins. By the way, this is gamma. It's my Greek letter gamma. And I'll tell you why they call them gamma globulins when I get to chapter 19 with you. So all these proteins. So proteins are a very diverse class of macromolecules.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. A hydrogen atom, and 4. A group that differs from one amino acid to another and is represented by the letter R.
Okay, before I go on, there's 20 or 21, depending on what discipline you're looking at. If you're in microbiology, there's 21 amino acids. If you're in human anatomy and physiology, there's 20 of them. And that means bacteria do something slightly different than humans, but that's not a big deal. There's 20, just think of 20 amino acids. Now, that means there's 20 different R groups because the rest of the amino acid is the same between amino acids, but there's 20 different R groups. And as you can see, when you hover over the R group, this box comes up over here, and you can read it, and it says, hey, some of these R groups are acidic, some of them are basic, some of them are neutral, some of them are nonpolar and hydrophobic, some of them are polar and hydrophilic. So we have different classes of R groups, and there's 20 different amino acids. Now, this narrator, she told you that proteins are long chains of these amino acids. How do I know which amino acid to put in which order? So how do I know to do glycine, alanine, threonine, leucine, isoleucine, 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 phenylalanine, tyrosine, 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 etc., whatever I do? And the answer is my DNA dictates it. So see, my DNA dictates what order to put the amino acids in, and therefore my DNA dictates what my proteins look like. And my proteins, of course, dictate what I look like and you look like. So indirectly, your DNA dictates what you look like because it controls which proteins are made. Dehydration synthesis of the 2 amino acids, 3 amino acids, 4 amino acids.
This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Okay, so that's dehydration synthesis of the protein monomers called amino acids into the polypeptide. Now, you put 2 monomers together, you get a dimer or a dipeptide. Put 3 amino acids together, you get a trimer or a tripeptide, 4 tetrapeptide, 5 pentapeptide, 6 hexapeptide, 7 heptapeptide, 8 octapeptide, 9 nonapeptide, 10 decapeptide. Well, when the heck is Kiggins going to call it a polypeptide? There's no hard and fast rule. I call it a polypeptide after 10 because I don't know what comes after decapeptide. But you might call it a polypeptide after 7 because you don't know octapeptide or nonapeptide. There's not really a hard and fast rule. These are the amino acids, and I know this picture's not in your book, but I know some of you guys are interested in things like this. At physiological pH, this is what the amino group looks like. It's an NH3 molecule or moiety. It's not a molecule, it's part of a bigger molecule. It's an NH3 moiety, and the N is positively charged, and the carboxylic acid group is deprotonated and negatively charged. This is what they look like at physiological pH, which if you don't remember, physiological pH is 7.35 to 7.45. So that's why the amino acids are drawn that way. All right, and these are all the amino acids, and you can see that they're non-polar hydrophobic amino acids, there's polar hydrophilic amino acids, there's acidic amino acids, and there's basic amino acids. The 2 that I mentioned previously, why in schnapps do I talk about sulfur? Sulfur is found in cysteine and methionine. That's where sulfur is found, cysteine and methionine. All right, so these are your 20 amino acids. And by the way, the yellow, green, and blue and pink groups are the R groups. I hope you realize that these yellow groups are the R groups. These green groups are the R groups. These blue and pink groups are the R groups. And by the way, you can see that we have 3-letter and 1-letter abbreviations for these amino acids. I have the 3-letter abbreviations memorized, but I do not have the 1-letter abbreviations memorized. At one time I did, I no longer do. Something you can look up, and you don't need to memorize them either. All right, proteins. Because they're hundreds to thousands of amino acids long, we talk about their architecture in a step-by-step fashion, and we say they have a primary structure, a secondary structure, a tertiary structure, and sometimes a quaternary structure. Not all proteins have a quaternary structure. What is the primary structure? It's just the amino acid sequence. So if you go back here to these amino acids and you say, well, what's the amino acid sequence? Now this is with replacement, right? So you could be tryptophan, 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 proline, glutamine, 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 glutamine. I mean, this is with replacement. So you have 20 amino acids. Tell me how long your protein is. Let's say your protein is 500 amino acids long. Well, there's 20 to the 500th exponent possibilities. Just think of it like this. I know some of you haven't had statistics yet. If you have 500, let's just say you have 5. Actually, let's say you have 3. Let's make this simple. Let's say you have a tripeptide, 3 amino acids long. In this spot right here, there's one of 20 amino acids. It could be any one of these over here. So in that spot right there, there's 20 different possibilities. In this spot right here, there's 20 different possibilities. Because remember, it's with replacement. You could use serine twice or 3 times. And in this last spot right here, there's 20 different possibilities. Overall, how many possibilities are there? But see, I don't have to run through 20 to the 3rd possibilities. My DNA dictates what order to put them into. So I have primary structure, which is just my amino acid sequence. I have secondary structure, which is either an alpha helix or a beta pleated sheet. And then I have a tertiary structure, which is the overall 3-dimensional shape. And then quaternary structure is when you have more than one polypeptide chain, all of them with tertiary structures combined. And with quaternary structure, it's sometimes, and we call the polypeptides subunits. So you have more than one subunit combined together to make the overall protein. So let's look at these structures. This is the primary structure right here. Primary. These are the 2 secondary structures right here. This is the alpha helix, and this is the beta pleated sheet, or what they're calling just the beta sheet. All right, this is the tertiary structure right here. It's the overall 3-dimensional shape. You can see it's folded, and I have a alpha helix in here, and I have some turns. And you can actually see a prosthetic group added in, which I haven't talked about yet, but I will. And then lastly, you might have quaternary structure, but not every protein does. And this is showing you that there's a polypeptide chain in dark purple. There's a polypeptide chain in dark purple, 2 different ones. There's another polypeptide chain in light blue. There's another polypeptide chain in light blue. And because the 2 blue ones are called alpha subunits, and the 2 purple ones are called beta subunits, what we say hemoglobin has is alpha 2 beta 2 architecture. Hemoglobin has alpha 2 beta 2 architecture. 2 alpha subunits, 2 beta subunits. Now, what the heck is this? These are called heme molecules because we're talking about hemoglobin. The heme molecule is a prosthetic group. Like if you lose your arm or leg, you have a prosthesis. Well, this is a non-protein prosthetic. This is a non-protein portion of the protein called the prosthetic group, but it's absolutely necessary for functioning of this protein. The prosthetic group comes in 2 flavors. It can be a cofactor or a coenzyme. And we'll talk some more about them in a few minutes. So a lot of proteins have these prosthetic groups that are non-protein portions added to it. So that's the architecture of a protein. Now, many proteins are enzymes, and most enzymes are proteins. Remember, there's a small number of enzymes that are ribozymes, RNA, but most of them are proteins. Enzymes speed up chemical reactions. They have a induced fit model where the substrates have to fit into the enzyme. They have to fit. After they fit in there, you can make or break bonds. In this case, you're making a bond. 2 substrates becoming 1 product, you're making a bond. But you could have an enzyme that breaks bonds, and 1 product would become one substrate would become 2 products. So in this case, we're actually doing dehydration synthesis. 2 substrates combine on the enzyme. The enzyme speeds up the reaction and pumps out product. So enzymes, most enzymes are proteins, and they can be catalysts and speed up reactions. Here's the cool thing about this enzyme. In here, the enzyme might change shape a little bit. As you can see, it just changed shape a little bit. You see a little dimple there that's not over here. But from here to here, the enzyme doesn't change, meaning it looks the same in the beginning as it does at the end. Does it transiently change shape in the middle? Yeah. But at the end, it looks just like it did in the beginning, and it's ready for another reaction. In other words, the enzymes aren't consumed. I don't have to keep making more and more enzyme. That one enzyme can run this reaction over and over and over again. They're not consumed in the reaction. Enzymes are catalysts. They speed up reactions. How they speed up reactions is by lowering the activation energy. Enzymes have these three characteristics. They're specific for substrates. You know, if you have alcohol dehydrogenase, ADH, alcohol dehydrogenase, it dehydrogenates alcohols. It doesn't act on other molecules other than alcohols, like ethylene glycol or isopropanol or ethanol or methanol. It has to be an alcohol. It doesn't act on a lipid, it acts on alcohols. If you have NADH dehydrogenase, it only acts on NADH. It doesn't act on other types of substrates. They're very specific. So enzymes have specificity. Enzymes have saturation limits. Let me go backwards a little bit. I could add, the more S1 and S2 I add here, the faster the reaction goes. Because as soon as the first S1 and S2 turn into product and leave, another one comes in and binds. Now, at some point in time, I've added enough S1 and S2 that the enzyme is completely saturated. As fast as it pumps out product, there's another S1, S2 going in. That's a saturation, and adding more substrate doesn't make the reaction go faster. That's called a saturation limit. So enzymes are saturable. I can saturate them, and adding more substrate doesn't increase the reaction rate at all. And lastly, and maybe most importantly, is enzymes are controllable. I can regulate them. And I haven't talked about allostery yet, but I'll talk about allostery and allosteric sites. And that's a major way we control enzymes. And you know what? When you talk about reactions, the metabolic pathways and things like that, yeah, it's great to go memorize a metabolic pathway, and you will in your life sometime. But it's not about that memorization of the metabolic pathway. It's about understanding how the pathway is controlled and how homeostasis is maintained. That's what it's about. Although memorizing does give you the information right at the tip of your tongue or right at the tip of your mind rather than looking it up all the time. All right, so let's look at how enzymes work.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
All right, so now is just chit-chat. I want to show you guys, tell you guys something cool. There's a thing, I'll actually do it from the video. There's a subdiscipline of biochemistry, or maybe protein chemistry maybe, called enzymology. Very, very cool topic. And this is just chit-chat. You don't need to know this for any test. But you see how these substrates are coming in, how they're coming into the active site. Well, some enzymes, it matters which substrate binds first. So this orange one might have to bind before this gray one does, or the gray one might have to bind before the enzymes, before the gray one might have to bind before the orange one. So if it matters which one comes in first, we call them uni-uni reactions. If it doesn't matter which one binds first, we call them bi-ping-pong reactions. And some enzymes are so complex and and have great quaternary structure, and there's even more than one active site going on. They're tandem enzymes, and they're huge, that we have enzymes called uni-bi-bi-ping-pong mechanisms, and that's just kind of it's really cool to read some of these enzymology books, and they talk about these mechanisms, and they sound kind of silly, or you might sound silly if you're reading aloud saying, oh yeah, it's an uni-bi-bi-ping-pong mechanism. So you guys can look that up if you're interested, but it's pretty cool stuff. Enzymology is a really cool topic. All right, we already mentioned this a little bit. The protein without any prosthetic group is called an apoprotein, just protein. And it may or may not be functional. If it needs a prosthetic group, it won't be functional without it. By the way, if it's the protein that's an enzyme, it's called an apoenzyme, just protein. Just protein, all right? And then if it needs a prosthetic group to be functional, and we add that prosthetic group to it, now we call it a holoprotein with prosthetic group and functional. So the holoenzyme or the holoprotein is functional, and it's the apoprotein or apoenzyme if it's lacking the prosthetic group. All right. Now, what do the prosthetic groups look like? Well, first of all, it's a cofactor if it's inorganic. Now, what organic means is contains carbon. What inorganic means is no carbon. So these are minerals. All right. And what a coenzyme is organic. So these guys are vitamins. So yeah, your vitamins and minerals are quite often cofactors or coenzymes for the enzyme. They have to make the holoenzyme by eating your vitamins and minerals. And in addition, we have isozymes, or sometimes called isoenzymes. And what that means is this. There's different versions of a given enzyme. So if you have an enzyme that breaks down ethanol to acetate, all right, let's say you have an enzyme that does that. It's called alcohol dehydrogenase. John Doe might make alcohol dehydrogenase 1, but Jane Doe might not have the genes for alcohol dehydrogenase 1, and she makes alcohol dehydrogenase 2. But both alcohol dehydrogenases break down ethanol to acetate. So they're both doing the same reaction, but they're not exactly the same protein. There's an ADH1 gene that codes for ADH1, and there's an ADH2 gene that codes for ADH2, and these are called isoenzymes. So, and you might have heard of some of these before. If you had a grandparent, great-grandparent that had a heart attack, you might have heard of them measuring a CKMB on your family member. That's an enzyme called creatine kinase MB. It's an isoenzyme because there's also a creatine kinase BB, and there's also a creatine kinase MM. But see, BB doesn't exist in heart muscle, and MM doesn't exist in heart muscle, but CKMB exists in heart muscle. So we measure CKMB to see if your heart muscle is damaged. CKBB is found in the brain, and CKMM is found in skeletal muscle. So we have isoenzymes and isoforms of proteins that we can use clinically to tell which tissue is being damaged. And that's pretty cool clinically.
This animation demonstrates how B vitamin coenzymes must be present in order to enable some enzymes to carry out their functions. Many of the B vitamins assist in the complex biochemical processes that produce energy from food. The B vitamins are essential parts of coenzymes. Coenzymes are part of the machinery that allows reactions to take place. Enzymes are large protein molecules that catalyze biochemical reactions by lowering activation energies. Enzymes accomplish catalysis by configuring chemical reactants in the best possible spatial arrangement for reactivity. This enzyme catalyzes the release of carbon dioxide when producing energy from carbohydrates. Combining with the coenzyme helps the inactive enzyme achieve the appropriate three-dimensional configuration. For this reaction, thiamine must be present as the coenzyme. The green coenzyme molecule above is thiamine pyrophosphate, the cofactor needed to permit the enzyme and the substrate, pyruvate, to combine in the correct orientation to catalyze the reaction. Now all three molecules are nested together, and the substrate, pyruvate, combines chemically with the coenzyme, thiamine. The biochemical reaction can take place, and energy, as ATP, and CO2 are produced as products of the reaction. Once this happens, the remaining complex, containing the two-carbon fragment, thiamine, and the apoenzyme, rearranges and releases the two-carbon fragment, acetaldehyde, and the ATP. The departure of the acetaldehyde opened the thiamine pyrophosphate to combine with another molecule of pyruvate, and the cycle continues. Meanwhile, the energy is now in the molecule ATP and is available to do work or to be stored. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate. Here you see the conformational change in the enzyme needed to configure the enzyme to accept the substrate in a lock and key complex. This is called an allosteric control. The cofactor approaches the inactive enzyme and bonds to it in an enzyme-coenzyme complex. The new configuration is a good fit for the substrate. Once this enzyme cofactor substrate complex is formed, energy can be released as ATP, and the other reaction products can be released from the complex. Now the correctly configured active site on the enzyme is free to engage another substrate. The amount of B vitamins that are needed to serve as coenzymes is quite small. There is enough thiamine activity in 1.2 milligrams to meet the average person's needs all day. Extra B vitamins will not produce more energy. Excess B vitamins in the body will pass out of the kidneys into the urine. This table shows some details about several of the roles played by B vitamins as coenzymes.
I like this table. And by the way, you know what upper level nutrition is? It's just biochemistry. I mean, I think nurses, if you're a nursing student, you have to take a nutrition class. But that's not the kind of nutrition class I'm talking about. They won't go into the detailed biochemistry of it. But master's level nutrition course is a lot of biochemistry. We already saw the thiamine in that video back there when it was a decarboxylation reaction. Riboflavin is necessary to make FAD, the electron carrier FADH2. Niacin is needed to make NAD and NADH, another electron carrier. Pyridoxine is involved in a whole bunch of aminotransferase reactions we won't talk about much. Pantothenic acid is pretty cool. It's needed to make a molecule called coenzyme A. So you'll see pantothenic acid at some point in time in your life. And then biotin is a carboxyl group carrier, so it's involved in carboxylation reactions. So those are some B vitamins. Oh, he said something I wanted to point out. Let me take this back. Right here, the thiamine is a cofactor. I'm sorry. It's a coenzyme. It's not inorganic. It's organic. Thiamine is a vitamin. It's organic. It's a coenzyme that binds in the active site. But he showed you that that's not the only way that has to happen. He said that you could also have a coenzyme that binds in an allosteric site. So this is the allosteric site right here. This is the active site. The active site's not active right now because it's the wrong shape. But as soon as your coenzyme gets in there, now your active site is the right shape. So these vitamins could be active site or allosteric site actors. Now, proteins can denature, and what that means is you unfold their quaternary or tertiary structure, and maybe even their secondary structure. And actually, I should say quite often their secondary structure. But the thing that denaturation doesn't do is it doesn't change the primary structure. It does not. When you hard boil an egg or fry an egg in the frying pan, the egg white that is translucent in kind of snotty consistency is egg white before you fry it, and it's egg white after you fry it. It's the same primary structured protein. But what you've done is you've unfolded it. You haven't changed the amino acid sequence at all. It's still the exact same amino acid sequence, but it doesn't have the same secondary, tertiary, and quaternary structure. You've unfolded it. Sometimes the unfolding is permanent, and sometimes the unfolding is temporary. So unfolding of proteins is called denaturation. And this is the very reason why fevers that are too high for too long are dangerous. Because if you have a fever that's too high for too long, it can lead to protein denaturation, especially in your brain, and that can lead to permanent brain damage. So here's a video about denaturation.
This familiar gelatin dessert actually is a good example of the process of coagulation of proteins into a three-dimensional latticework that entraps water molecules to produce a semi-solid gel. This animation explains the physical chemistry behind the process. Proteins are synthesized by polymerizing amino acids. The polymerization occurs by repeatedly forming peptide bonds that link individual amino acids together into a chain. There are three structural features that influence the three-dimensional shape of a water-soluble protein. Primary structure is the peptide bond between individual amino acids that creates a long chain of connected amino acids. These long chains of polymerized amino acids have hydrophobic, water-repelling, and hydrophilic, water-attracting projections that are oriented perpendicular to the chain, as shown in this illustration of a growing protein chain. Secondary structure is the helix that the protein chain curls into as a result of hydrogen bonds and other weak forces. Tertiary structure is created when the protein molecules fold back on themselves outside of the helical segments to put the hydrophobic portions to the interior and the hydrophilic portions to the exterior. Several helical regions can exist in different portions of the molecule. When the protein has folded and refolded to reach its most stable configuration, it will have mostly hydrophilic amino acid residues on the exterior and mostly hydrophobic residues directed into the interior. When natural proteins are subjected to physical or chemical treatment, their structure changes and they become unnative or unnatural. We call that process denaturation. In this example, heating the proteins in solution imparts energy to the protein molecules. This added energy is enough to break the relatively weak forces that hold the protein in its refolded and helical tertiary and secondary configurations. As the process of denaturation proceeds, the protein molecule unfolds more and more, and the internally directed hydrophobic regions now become exposed on the outside of the molecule. The peptide bonds are largely hydrophilic. Once these segments are set free from each other, they attract water molecules. The recruitment of water molecules entraps the water molecules in close proximity to the protein strands. The hydrophobic portions of the molecules are also exposed. This situation is unfavored because the hydrophobic portions of molecules are not stable in an aqueous environment. Hence, upon unfolding, the hydrophobic regions on individual protein molecules will associate with hydrophobic regions on other protein molecules. This situation encourages the association of these protein molecules into larger and larger random three-dimensional structures. The molecules aggregate into very large water-insoluble collections that are quite randomly assembled. As the proteins denature, latticework structures grow amorphously and attract the solvent water molecules into cell-like structures. The self-associated water molecules, in groups, adhere to the surface of hydrophilic regions of the protein, while hydrophobic regions of the protein dissolve into each other and provide the energy to retain the structure. As this process continues irreversibly, all of the protein molecules are recruited to this large insoluble mass in a randomly organized structural framework that contains entrapped water molecules. One example of the consequences of unfolding and reassociating protein molecules is coagulation of egg white. Frying an egg is no more complicated than denaturing the egg white protein. The assembly of irreversibly denatured protein molecules results in formation of a solid gel. The gel entraps water molecules inside the white into a semi-solid structure which holds its shape under normal conditions. Other examples of denatured protein assembling into three-dimensional structures include the baking of yeast-risen bread, coagulation of meat proteins by cooking in such products as hot dogs, and in the solidification of gelatin upon cooling of a solution. Denaturation and coagulation of proteins is a complex, irreversible process, but the study of denaturation has allowed us to better understand the three-dimensional structure of native proteins.
All right, the key concept there is denaturation does not change the primary structure. It only changes the folding, the secondary, tertiary, and potentially quaternary structure if that exists. I want to show you a couple mistakes in this video. Here's one mistake. That egg would be perfect if it had some pepper on it. That's one mistake. And here's another mistake. That gelatin right there would be perfect if there was some grapes and some bananas in there. I don't know who makes gelatin without fruit in it, but that's just crazy. That's a mistake. So those two food products were not made well. I just wanted to point out those mistakes to you. That's it. Next topic is nucleic acids.